protein cow. So we're going to feed our protein cow some protein. And in the rumen, we're going to fractionate that protein into really two piles, two fractions. The first fraction would be DIP, which would be degradable intake protein. We also said this might be referred to by some people as RDP, or ruminal degradable protein. And then the other fraction we'll put all the way down here at the bottom so we don't have to think about it again while we're in the rumen, and that will be UIP, or undegradable intake protein. You might see this referred to as RUP, rumen undegradable protein. Another term you might see more commonly would be escape protein. And basically, they're calling it escape because it's escaping the rumen intact. And if we want to provide a specific amino acid to the ruminant animal, we would need to get it in the UIP fraction as escape protein. We look at DIP. It gets further fractionated. You could feed lots of different sources of DIP. And DIP, again, is degradable intake protein, meaning the microbes can degrade it, they have access to it. And degradable intake protein can be fed as feed protein, feed peptides, we have feed amino acids, we have feed ammonia, and we have feed urea. And basically, the microbes are going to degrade feed protein to feed peptides to feed amino acids to feed ammonia. And in the opposite direction, feed urea can be hydrolyzed into feed ammonia via microbial activity as well. And so the enzymes that make this possible are all of microbial origin. And so we've been abbreviating those as BE, indicating they're of microbial origin. Okay, and a real important step is as we take feed amino acids and make them into feed ammonia, we're going to release two things. The first thing would be a carbon skeleton. And generally we've been abbreviating carbon skeleton CS. And then the microbes are also going to derive or get some ATP from this process. So we're getting a carbon skeleton, and we're also getting ATP for the microbes. Important thing to keep in mind about this carbon skeleton is this carbon skeleton is also a VFA, and VFAs get absorbed, as we learned last time, and can provide energy to the cow. Okay, and when they get absorbed, they go to the blood, through the blood to the liver. Okay, the feed ammonia can enter the ammonia pool, and there's one other source for this ammonia pool, and that would be urea that crosses the rumen epithelium from the blood, contributes to the ammonia pool. And this occurs when we're breaking down either urea, whether it's feed urea or endogenous urea. So you might want to think of this as endogenous urea because it's coming from the animal. When we're breaking those down, or creating ammonia, that's the result of urease. And urease is an enzyme that comes from the ruminal microbes. Okay, so now what's going to happen is the microbes are going to take this amino acid, or this ammonia, and synthesize microbial amino acids. And to do this requires ATP. They're going to use energy to do this. And it also requires the addition of a carbon skeleton. Okay? While we're here, this reaction can also proceed the other direction, and they will produce a carbon skeleton, and they will create or release ATP for them to utilize. Microbial amino acids can also come directly from feed amino acids. In class, I think we said bacteria like Streptococcus bovis, or your non-structural carbohydrate utilizing bacteria, really prefer to get preformed amino acids, whereas your fiber fermenting bacteria prefer ammonia. And then the microbes will synthesize microbial peptides, and that's going to require energy. And then microbial peptides ultimately become microbial protein, again requiring energy. Okay, and we really want this energy for the synthesis of microbial protein to come from fermentation of carbohydrates. So down here in the rumen we'll write fermentation of carbohydrates provides energy for MCP synthesis, or microbial crude protein synthesis. And much in the same way, carbohydrates provide energy for microbial crude protein synthesis. The protein that the microbes are synthesizing is what's responsible for digesting and fermenting the carbohydrates we've fed to the cow. Okay? And so this microbial protein leaves the rumen as microbial crude protein. And in the abomasum, microbial crude protein and UIP basically get utilized about the same. Really an important thing to keep in mind is the relationship between carbohydrates and protein, and how microbes require energy from carbohydrates to synthesize protein, and they require protein to be able to ferment and utilize the carbohydrates. So for an effective rumen, you have to have a balance between crude protein and available energy. Really more specifically than crude protein, you need to have a balance between DIP and fermentable energy. In the abomasum, these two are going to become oligopeptides. And short peptides. And that's going to be the result of the action of two things. The first thing will be two secretions. It's better than thing. Two secretions. The first secretion would be HCl, which works on the secondary, tertiary, and quaternary structures of protein. So it's going to denature the protein. And then the other secretion that will play a role in creating those oligopeptides and acidifying the digester. Really, this is what's creating the oligopeptides, would be pepsinogen. So pepsinogen is secreted. It's inactive as pepsinogen, but in the stomach it becomes pepsin. Down here under our rumen, we might capture how that occurs. So again, the animal is going to secrete the inactive zymogen, pepsinogen. And we want to secrete inactive zymogens because proteolytic enzymes aren't necessarily good for the long-term sustainability of a organ, because they're made of proteins and they're going to digest. Organs are made of proteins, and proteolytic enzymes would digest the organ, which would be bad. So we end up with pepsin, which is the active form. It can be activated by HCl or pepsin, but the initial activation is the result of HCl. Okay, so what we have moving to the small intestine is the products of protein digestion. Real quick, just remember these zymogens are going to break the primary structure of the protein, so they're actually going to break the peptide bonds. So we have the products of protein digestion, which would be oligopeptides, short peptides, and then acidified digesta, which is going to act on two cell types, the S cells and the I cells, which are both located in the proximal duodenum, and they are going to produce two products. I cells will produce CCK, and S cells will produce secretin. This is going to act on the pancreas to produce two things, two secretions, Na plus HCO3 minus. And while we're doing that, we'll just go ahead and show the small intestine also producing that, producing the buffer to raise the pH. And the other secretion coming from the pancreas as a result of secretin and CCK would be zymogens. CCK and secretin are also going to cause the small intestine to release enteropeptidase. Okay, so back under the rumen, we are going to write pancreatic zymogens. And our pancreatic zymogens are trypsinogen. Okay, chymotrypsinogen. And last but not least, procarboxypeptidase. Okay, all three of these are secreted as inactive enzymes, and trypsinogen will be first activated by enteropeptidase. Which is a clever system for taking an inactive enzyme in the small intestine and activating it in the small intestine. Once activated, trypsin can become self-activating. So a trypsin can then activate another trypsin to trypsin. And then trypsin is going to activate the other two as well. And this is why the trypsin inhibitor is important. Less important in the ruminant because the rumen's going to take care of some of it, detoxify it, make it inactive, so we don't have to worry about it as much. But if you don't have active trypsin, you're going to decrease protein digestion because these pancreatic zymogens can't be activated. So chymotrypsinogen becomes chymotrypsin, procarboxypeptidase becomes carboxypeptidase A and B. And now they're free to carry out protein digestion in the small intestine. Okay, we have 3 other enzymes that are secreted by the small intestine. And those 3 additional enzymes would be amino peptidase, dipeptidyl amino peptidase, and last but not least, tripeptidase. And all 3 of these, just like every other enzyme we've listed today, is going to break peptide bonds and allow us to create the 3 products that are absorbed. And the 3 products absorbed from the small intestine are amino acids, dipeptides, and tripeptides. Okay, so we'll stick an enteroside here. This is our enterosite. We have 2 types of transporters for amino acids. We have sodium dependent, which is going to require or use sodium. And we have sodium independent, which doesn't use sodium, so relies solely on the concentration gradient of the amino acids. And then for peptides, the di and tripeptides, we use pept1. And when we do that, we're going to co-transport a peptide and a proton. So the cell is going to want to get rid of that proton, so it will exchange that proton for a sodium. Wrong arrow. The arrow is going into the cell on this one. Make it not look arrowish. So we're going to exchange the sodium for the proton. So the sodium dependent and the pept are dependent on potassium, on the sodium-potassium pump, to keep the sodium concentration low inside the cell. That's an ATP right there. It's hard to see, but an ATP, I promise. Not much better, but ATP. Okay, and then the last transporter we have on the cell is the amino acid transporter, AAT, and that is going to facilitate the bidirectional transport of amino acids. And inside this enterosite, we have all amino acids because there's a peptidase that cleaves di and tripeptides into amino acids. And those amino acids are going to go to the liver. Okay. And depending on what the animal's doing, if it's a growing animal, it would be depositing muscle. If it's a pregnant cow, it might be depositing, it might be growing a calf, a fetus. So if amino acids are in excess versus amino acids are deficient, if they're in excess, the cow will be creating tissue. We're going to label that as muscle. Something to kind of keep in mind is like a pregnant cow will mobilize her own tissue to support the growth of the fetus and to meet her maintenance amino acid requirements. So she'll actually mobilize herself. So a certain tissue type might be in negative amino acid balance to support lactation or gestation, or just to meet the maintenance requirements of that animal. If in contrast we just have excess amino acids, or the animal is needing these amino acids, those are broken down into ammonia, and they're also broken down into a carbon skeleton, which the animal can use to synthesize glucose, because some amino acids are gluconeogenic, meaning you can synthesize glucose from them, or she might just use them for energy. Okay, and this is true in you as well. If you consume excess amino acids, you're going to use those amino acids as a source of energy to meet your glucose requirements for your brain and other essential tissues. It's going to result in the production of ammonia, but we'll take care of that in a second. This is how, if you fast for a really long time or don't eat right, you begin to lose muscle mass because your body's trying to meet its protein requirements for maintenance as well as its glucose requirements. It's also how Atkins works, or the keto diet works, is basically you're trying to, or you are synthesizing glucose from other sources. So ammonia is toxic. So in the liver, we're going to produce urea, or the cow is going to produce urea. We produce urea as well. And that urea can exit the body in the urine. It can also be recycled back, and so that's where this endogenous urea shows up. Okay. And in being recycled back, it could also be recycled back through the mouth. So in the saliva, they can produce, or in that saliva secretion, you can create urea, or urea can be secreted, go into the rumen, be hydrolyzed as an additional source of ammonia. The last place urea could be recycled to, it's recycled to the entire gastrointestinal tract, but from a useful standpoint, the last place that we might think about sending it would be to the large intestine. So we have urea here. Inside the large intestine, because there's microbes that produce urease, we end up with ammonia. That ammonia could be reabsorbed. If that ammonia is reabsorbed, it goes, we'll make it a different color. Maybe it'll make it less confusing. That ammonia that gets absorbed goes back and is used to synthesize urea. Okay, we'll do this slowly. The same thing can happen from the ammonia pool in the rumen, in that if we have excess ammonia, it can be absorbed and go back to the liver, or go to the liver and be synthesized to urea, and can be recycled again and again and again and again. Okay? That's when you have high ruminal ammonia concentrations. We learned in carbohydrates, when we talked about non-structural carbohydrates, that they decrease ruminal ammonia concentration because the microbes are growing. The microbes can synthesize lots of microbial crude protein because they have a lot of energy. So you'd expect the ammonia pool to be less. So you'd expect less ammonia to be absorbed. You'd also expect maybe more urea to be recycled because there's less ammonia. In the large intestine of the cow, we can take and break feed proteins as well as endogenous proteins. The microbes can break them down, and we can end up with amino acids. And those amino acids could be further broken down into ammonia and the carbon skeleton. And the microbes would get ATP from that process. This carbon skeleton, or VFA, could be absorbed and then go to the liver and become a source of energy for the animal. They could also use it, plus some ammonia, to synthesize MCP. Or we could directly take this amino acid and use it for MCP, and that gets excreted in the feces. Okay, but any digestion that's going to occur is a result of microbial activity, and that could be digestion of feed protein as well as endogenous proteins. Okay. Kind of trying to think about anything we want to try and summarize. So the products of protein digestion absorbed in the large intestine would be ammonia and VFAs. The products absorbed from protein digestion in the small intestine would be amino acids, dipeptides, and tripeptides. The products absorbed in the rumen would be ammonia and VFAs. And kind of the last thing we want to think about before we wrap this drawing up, which has taken a really long time, sorry about that, is that microbes moderate protein quality. Okay, when we're talking about, and what I really mean is they can improve the amino acid profile of a low quality protein, corn, but in contrast, they would decrease the protein quality of a high quality protein. So they would decrease the amino acid profile of a high quality protein. So if you gave them milk or soybeans, the amino acids that the cow receives in the small intestine would be less effective at addressing her requirements than the actual protein you fed. In contrast, if you feed them a low quality protein, say corn, which has a poor amino acid profile for supporting growth, the amino acids that the cow is going to absorb or that show up at the enterocyte are going to be better capable of meeting her amino acid requirements. Okay, so that completes the cow. Hopefully this is helpful.